Could measuring protein clumps in our cells be a new way to find out our risk of getting age-related diseases? Professor Dorothy Dorman and Professor Edward Lemke of Johannes Gutenberg University Mainz JGU, who are also adjunct directors at the Institute of Molecular Biology IMB in Mainz, proposed the concept of a protein aggregation clock to measure aging and health in a new perspective article published in Nature Cell Biology. As we age, the DNA and proteins that make up our bodies gradually undergo changes that cause our bodies to no longer work as well as before. This in turn makes us more prone to getting age-related diseases such as cardiovascular disease, cancer, and Alzheimer's disease. One important change is that the proteins in our cells can sometimes become misfolded and clumped together to form aggregates, so-called amyloids. Misfolding and aggregation can happen to any protein, but a specific group of proteins known as intrinsically disordered proteins, IDPS, are especially prone to forming amyloids. IDPS make up around 30% of the proteins in our cells, and they are characterized by having no fixed structure. Instead, they are flexible and dynamic, flopping around like strands of cooked spaghetti. While the molecular mechanisms are widely debated and an important aspect of basic research, scientists know that aggregates formed from IDPS tend to accumulate in many long-lived cells, such as neurons or muscle cells, as we age. Moreover, they can cause many age-related diseases, particularly neurodegenerative diseases such as Alzheimer's and Parkinson's disease. Thus, having many aggregates in a cell could be an indicator of how unhealthy the cell is or if a person is likely to develop an age-related disease soon. In their recently published article, Dorman and Lemke propose that IDP aggregation could be used as a biological clock to measure a person's health and age. If developed further into a sensitive diagnostic test, a protein aggregation clock could be extremely useful. Firstly, doctors could use it to help diagnose age-related diseases at very early stages or identify people who are not yet sick but have a higher risk of developing disease as they age. This would allow them to be given preventative treatments before they develop severe disease. Secondly, scientists could use it to assess the effects of new experimental treatments to reduce protein aggregation in order to prevent or delay age-related diseases.